Welcome to another episode of Question of the Day. So, Dr. Dell, a 4-year-old child presents with fever and signs of meningitis. Cerebrospinal fluid culture grows a gram-negative coccobacillus, suspected to be Haemophilus influenzae type B. The organism shows minimal growth on sheep blood agar alone. However, when the agar is cross-streaked with Staphylococcus aureus, there is enhanced bacterial growth around the streak. What mechanism best explains this phenomenon?
All right, so the organism that is cultured here is Haemophilus influenza type B, and it's been cultured on a sheep agar together with Staphylococcus aureus. So what you have to know is that if only Haemophilus influenza is cultured on a sheep blood agar, there's going to be minimal to no growth. The reason is that the Haemophilus influenza will need certain factors for growth. And these factors, there are two. We have the V factor, also known as NAD+, then the X factor, also known as hemin. On the sheep blood agar, the red blood cells contain both of these factors. That is the V factor, NAD+, and the X factor corresponding to hemin. For the hemin to function, it just needs to be in the red blood cells. It just needs to be in an intact red blood cell. But for the NAD+, which is the V factor, to function, it needs to get out of the red blood cell, meaning you need the red blood cells to lyse or break down for that NAD+ to promote the growth of the Haemophilus influenza. So the sheep blood agar has certain factors that inactivate the V factor, that's NAD+. That enzyme is known as NADase, which prevents the functioning of NAD+, that's the V factor. Moreover, Haemophilus influenza is not able to cause the breakdown of the red blood cell, and it's not a beta hemolytic bacteria. So the NAD+ remains in the intact red blood cells, gets inactivated by the NADase, which is produced by the sheep agar blood, and without the V factor, which is the NAD+, getting out of the red blood cells, the Haemophilus influenza type B will not be able to grow. When this Haemophilus influenza is cross-streaked with Staphylococcus aureus on a sheep blood agar, what is going to happen is that the Staphylococcus aureus is a beta hemolytic bacteria, meaning that it causes complete hemolysis of the red blood cells. And the Staphylococcus aureus has sufficient amount of the X factor, which is hemin, and the V factor, which is NAD+. For the fact that Staphylococcus aureus causes beta hemolysis, that's complete hemolysis of the red blood cells, the NAD+ ends up getting released from the breakdown of the red blood cells and then promote the growth of Haemophilus influenza type B. So with that, you will see the beta hemolysis being caused by the Staphylococcus aureus, that's the complete hemolysis of the red blood cells, and around the Staphylococcus aureus growth, you will see the Haemophilus influenza type B also growing. That phenomenon is what is referred to as the satellite phenomenon.
Wow, interesting. We meet again, Dr. Dell. Thank you.